Good morning, my dear students. Today we are going to discuss about aerobic respiration. That aerobic respiration has four stages. One is glycolysis, second one link reaction, the third one Krebs cycle, fourth one electron transport system. In the anaerobic respiration, glycolysis is a common followed by fermentation, but in the aerobic respiration, after glycolysis, there is a link reaction, Krebs cycle, electron transport system is there. Glycolysis takes place in the cytoplasm of a cell, common in both aerobic and anaerobic. Krebs cycle link reactions takes place in cytoplasm and mitochondria of a matrix. Krebs cycle takes place in matrix of mitochondria. Electron transport system takes place in cristae of inner membrane of mitochondria. After this introduction, we can try to understand the meaning of glycolysis. It is a Greek word. The meaning consists of two words like is glykis as well as lysis. Glykis means sugar, lysis means breakdown. The glycolysis means breakdown of sugar molecules. That is, one glucose is breakdown into two pyruvic acids called as an glycolysis process. It is a common process in both aerobic as well as anaerobic respiration. This glycolysis process was discovered by three scientists. They are named as Emden, Meyerhoff, and Parnas. The first scientist name is Emden, second one is Meyerhoff, the third one is Parnas. The glycolysis process have the 10 series of reactions. The 10 series of reactions was discovered by these three famous scientists. Hence, the glycolysis path is also called as EMP pathway. Because of based on the respect of the scientist's name, the pathway of the glycolysis is also called as EMP pathway. In the glycolysis, 10 series of reactions are participating. It takes place in the cytoplasm of a cell. In this glycolysis, sucrose is a raw material. The sucrose is converted into glucose plus fructose in the presence of invertase enzyme. This question also was asked in the previous NEET examination. So, which enzyme is used for the conversion of sucrose into glucose and fructose? That is invertase. And in this, glucose undergoes two series of reactions. That is a glycolysis followed by link reactions, followed by Krebs cycle, followed by electron transport system. Glycolysis is a first step in the respiration, that is aerobic respiration as well as anaerobic. In the glycolysis process, one glucose molecule breaks down into two pyruvic acids. That one glucose consists of 6 carbon, but it is the end of this glycolysis. Two pyruvic acids are formed. Each pyruvic acid consists of only 3 carbon. Mean one 6 carbon compound is converted into two 3 carbon compounds. It is occurs in the series of reactions of 10 series of reactions takes place here. Thus, each reaction takes place in the presence of enzyme. Several intermediates have been formed, but end product is only two pyruvic acids. In the series of reactions, energy also have been produced. That is, energy is produced in the form of ATP as well as in the form of NADH2. Here, only then glucose is undergoes. The consumption of oxygen is not at all possible. Only then some water molecules have been produced. Means consumption of oxygen is not takes place here. Hence, glycolysis takes place in the aerobic as well as anaerobic respiration. My dear students, now we can try to understand that is in glycolysis process. In this process, there is a 10 series of reactions just now we discussed. The first reaction is called phosphorylation, that is the addition of phosphate. The second one is called isomerization, then conversion of glucose into fructose. The third one is called phosphorylation, means addition of phosphate. Fourth one is cleavage, splitting. 6 carbon compound is splitting into 3 carbon compounds of dihydroxyacetone phosphate as well as glyceraldehyde 3 phosphate. And then fifth one is isomerization. Then glyceraldehyde 3 phosphate undergoes to next series of reactions, but dihydroxyacetone phosphate is converts into glyceraldehyde 3 phosphate to do the next series of reactions. In these 5 stages, glucose is converts into 3 carbon compound of phosphoglyceraldehyde 3 phosphate. Now we can discuss in detail. It is little bit of tough and little bit of twist topic. Just you can listen very careful and try to understand this. In these reactions, we can start with first, that is a glucose. That first reaction is, just we already discussed, there is a 10 series of reactions. That first reaction is called phosphorylation, that is addition of phosphate. Glucose converts into glucose 6 phosphate. For this glucose, phosphate is added from ATP, means glucose plus ATP gives rise to glucose 6 phosphate plus ADP. Glucose also consists of 6 carbons. Glucose 6 phosphate also consists of 6 carbon, but extra phosphate is added. That phosphate is given by ATP molecule. This reaction takes place in the presence of hexokinase enzyme along with magnesium ions. This enzyme question was asked in the previous. Which enzyme is used for the conversion of glucose to glucose 6 phosphate? Hexokinase. The second reaction is called isomerization. Glucose converts into glucose 6 phosphate converts into fructose 6 phosphate. In this addition of phosphate or removal of the phosphate does not take place. Only glucose molecules are converted into fructose molecules. Mean glucose 6 phosphate having the 6 carbon is converted into fructose 6 phosphate by having the 6 carbon. No changes in the number of carbon, only that changes is glucose to fructose. This conversion is called isomerization. This conversion takes place in the presence of enzyme is called phosphohexoisomerase enzyme is used. Then third reaction is phosphorylation, like a first step, addition of phosphate to fructose. Fructose 6 phosphate plus ATP gives rise to fructose 1,6 biphosphate plus ADP. Means one more phosphate is going to add for fructose 6 phosphate. Results, fructose have the 2 phosphates. One phosphate is attached at the first carbon. The second phosphate is already attached at the 6th carbon. Now the fructose has a 2 inorganic phosphates. One is at the first carbon, one is at the 6th carbon. Along with then ADPs are released out. The number of carbons in the fructose 6 phosphate is 6, as well as number of carbons present in the fructose 1,6 biphosphate also 6. Only changes is the number of phosphates present in the fructose phosphate is 1, that is the 6th one, 6th at the end of this reaction. Fructose has a 2 phosphates, one is at first one is at 6th one. This conversion takes place in the presence of an enzyme is called phosphofructokinase. Phosphofructokinase. The fourth reaction is cleavage, means splitting. Now fructose 1,6 biphosphate going to split as 3 carbon compounds. The fructose 1,6 biphosphate has a 6 carbon. It's a 6 carbon compound. It is going to split into 3 carbon compounds of 2. One 3 carbon compound is glyceraldehyde 3 phosphate or 3 phosphoglyceraldehyde. One more name for the glyceraldehyde 3 phosphate. The second one is dihydroxyacetone phosphate. The fifth reaction is isomerization. Then dihydroxyacetone phosphate is converts into glyceraldehyde 3 phosphate. Dihydroxyacetone 3 phosphate also has a 3 carbon, and also glyceraldehyde 3 phosphate also has a 3 carbon. But in this glyceraldehyde 3 phosphate form of 3 carbon compound undergoes to next series of reactions. That's why dihydroxyacetone phosphate also converts into glyceraldehyde 3 phosphate, called as isomerization. Now we can try to understand the next 5 series of reactions. In that, the sixth reaction is oxidation and phosphorylation. That is an phosphoglyceraldehyde 3 phosphate is converts into 1,3-diphosphoglyceraldehyde. This conversion takes place by the production of energy, that is NAD is phosphorylized or oxidized into NADH plus 2H plus. Means energy have been produced here, 2 NADH plus 2H plus are produced because of 2 moles of phosphoglyceraldehydes converts into 2 moles of 1,3-diphosphoglycerides. For each mole of phosphoglyceraldehyde, 1 mole of NADH plus H plus is produced. Hence, the 2 moles of NADH plus 2H plus are produced. This conversion takes place in the presence of enzyme is called glyceraldehyde phosphate dehydrogenase. This conversion is called oxidation and phosphorylation. Two are participating here. The seventh reaction is dephosphorylation, or here phosphorylation 1. Here the removal of phosphate from this 3 carbon compound called as a dephosphorylation. The removed phosphate, inorganic phosphate, is going to attach with ADP called as a phosphorylation. In the sense dephosphorylation, or we can call it as a phosphorylation. 1,3-diphosphoglycerate is added with, means react with 2 adenosine diphosphate, then results 3 phosphoglycerate is formed along with the 2 ATP molecules. This conversion also takes place in the presence of enzyme is called phosphoglycerate kinase. Mean each 3 carbon compound can produces 1 ATP molecule, hence results 2 ATP molecules are produced. In the glycolysis, it is a first reaction, first phosphorylation for the formation of ATP, hence called as a phosphorylation 1. Then eighth reaction is isomerization or intramolecular shift, means 3-phosphoglycerate is converts into 2-phosphoglycerate in the presence of phosphoglyceromutase enzyme. Means actually carbon is present, when phosphate is present at the third carbon of the phosphoglycerate, but that phosphate is shifting from third carbon to second carbon, hence called as an 2-phosphoglycerate. Then reactant also have the 3 carbons, as end product also have the 3 carbons. Only the phosphate position is shifting from third carbon to second carbon, hence it is called as an isomerization. The ninth reaction is dehydration. Two molecules of water is removed, means produced. The two molecules of water is removed from 2-phosphoglycerate, means 2-phosphoglycerate by losing two molecules of water, it is converts into 2-phosphoenol pyruvate. 2-phosphoenol pyruvate conversion is takes place. This is a takes place in the presence of enzyme is called as enolase. Enolase. Dehydration in the sense removal of water molecule. In this also energy is not produced. In the isomerization also energy production or consumption does not take place. The 10th reaction is dephosphorylation. It is the last reaction, last step of this glycolysis. It is also dephosphorylation, or we can call it as a phosphorylation 2. Means first phosphorylation taken place at the time of 1,3-diphosphoglycerate into then 3-phosphoglycerate conversion, 2 ATP molecules are produced. But the last phosphorylation, there is a second phosphorylation, or we can call it the dephosphorylation. In this dephosphorylation, 2 moles of 2-phosphoenol pyruvate is converts into 2 moles of pyruvate or pyruvic acid. This conversion takes place by the production of ATP form of energy, means 2 phosphoenol pyruvates plus 2 ADP molecules gives rise to 2 pyruvic acids plus 2 ATP molecules. This conversion takes place in the presence of pyruvate kinase enzyme. Few of NEET model questions from the today discussed topic. That is, question number 1 is the enzymatic machinery required for partial breakdown of glucose into 2 molecules of pyruvic acid without the involvement of oxygen is present in. Option 1 is given as aerobic prokaryotic only. Option 2 anaerobic prokaryotes only. Option 3 anaerobic prokaryotes and eukaryotes only. Option 4 all aerobic and anaerobic organisms. This out of this given 4 options, option 4 is the right one. All the aerobic and anaerobic organisms have the glycolysis process without oxygen consumption. The second question is how much energy is released in anaerobic respiratory by the incomplete oxidation of one molecule of glucose. It is a question based on anaerobic respiration. What is the amount of energy is produced at the end of anaerobic respiration by the incomplete oxidation? In the sense, not complete oxidation, means no Krebs cycle and electron transport. So option 1 is given as 52 kilocalories, option 2 is given as 56 kilocalories, option 3 is given as 15 kilocalories, option 4 is given as 16 kilocalories. Out of this one, option 2 is right one, that is a 56 kilocalories of energy. But in the aerobic respiration, 686 kilocalories of energy is produced, but in anaerobic respiration, 56 kilocalories of energy is produced. Then the question number 3 is that instant source of energy. The most common respiratory substrate is either aerobic or anaerobic. Option 1 is given as a glucose, option 2 is given as a fructose, option 3 is given as a sucrose, option 4 is given as all. But in the aerobic as well as anaerobic, the common respiratory substrate is glucose, means option 1 is correct one. Question number 4 is alcohol is produced during the process of. Option 1 is given as photosynthesis, option 2 is given as aerobic respiration, option 3 is given as anaerobic respiration, option 4 is given as none of these. Means alcohol is the end product of anaerobic respiration. Along with alcohol, lactic acid also end product of anaerobic respiration. Photosynthesis end product is starch. The aerobic respiration end product is carbon dioxide, water as well as energy. But in anaerobic respiration, alcohol is end product. Along with the alcohol, carbon dioxide end product. So option 3 is the right one. We can see that few of the questions asked in the previous NEET exams from the discussed topic. Then questions asked from the previous NEET examination. The question number 1 is, which of this following statement is incorrect regarding the fermentation? The options are given here. Propionibacterium is used to ferment the cheese. Option 1 is given as, but option 2 is given as the puffed up appearance of dough as due to the production of carbon dioxide gas. Option 3 is given as fermentation in mussel produces ethanol. Option 4 is given as toddy is made by fermenting sap from the palm. Here the question is asked incorrect one regarding the fermentation. So out of these four, third one is that incorrect. Fermentation in the mussel cells produce alcohol, ethanol? No. It can produces lactic acid. Alcohol is produced in the yeast cells. If you can see, the first option is correct one, as because of propionibacterium is used to ferment the cheese, which is a product of lactic acid, that is which is used edible. Second option is the puffed up appearance of dough as due to this production of carbon dioxide gas. This statement also correct one. At the time of eating bread, you are finding small pores in between. The pores are nothing but due to releasing of carbon dioxide. So then third option is fermentation in the mussel cells produces ethanol. In the mussel cells, inadequate of oxygen or lack of oxygen or insufficient of oxygen. Sometimes anaerobic respiration is going to take place. The second stage of anaerobic respiration, fermentation. The end of this lactic acid fermentation, lactic acid is produced. That's why after long running or playing cricket, something exercises, we are getting pain in the muscles like a burning due to that excess amount of lactic acid produces. Option fourth one, toddy is made up of fermenting sap from the palm. Toddy in the sense generally white water we can call as in, which the traditional people are drinking, especially in the rural areas, people drink the toddy. The question number 2 is, glycolysis is found in the cytoplasm of virtually all types of aerobic and anaerobic cells. In this process, glucose is converted into compound which is a common end product of glycolysis in aerobic as well as anaerobic. That is the meaning of question. Options are given here. PEP, phosphonol pyruvic acid. Option 2 is given as acetyl coenzyme A. Option 3 is given as pyruvic acid. Option 4 is given as citric acid. PEP is a product in the photosynthesis process, especially dark reaction, that is in C4 plants. Acetyl coenzyme A, there is a linking compound in between glycolysis and Krebs cycle. Then pyruvic acid is a 3-carbon compound, is the end product of glycolysis, both in aerobic as well as anaerobic. So fourth is the citric acid. Citric acid is product of Krebs cycle. So answer 3 is the right one, pyruvic acid. Now we can see the third question. Then question is the conversion of glucose into the glucose 6-phosphate. The first irreversible reaction of glycolysis is catalyzed by, means then glucose is converted into glucose 6-phosphate. It is the first one It does not convert then glucose 6 phosphate into the glucose. That is then which enzyme is useful for here? Option 1 is given as aldolase. Option 2 is given as hexokinase. Option 3 is given as enolase. Option 4 is given as phosphofructokinase. So the first action in the glycolysis takes place with the help of hexokinase enzyme. So option 2 is the correct one. So we can see the question number 4 from the previous NEET question paper. What is the role of NAD plus in cellular respiration? NAD plus, NAD in the sense nicotine, amide, adenine, dinucleotide. So options are given as option 1, it is a nucleotide source of ATP synthesis. Option 2, it functions as an electron carrier. Option 3, it functions as an enzyme. Option 4, it is the final electron acceptor for anaerobic respiration. Out of these 4, option 2 is correct one. It is functions as an electron carrier. But in photosynthesis, NADP acting as electron carrier. NADP in photosynthesis, electron carrier, electron acceptor. NAD plus electron acceptor, electron carrier in respiration. Means NADP full form is nicotine, amine, adenine, dinucleotide, phosphate. So here option 2 is correct one. From today's discussion, we have completed and we started the respiration. In that introduction of the respiration, in the definition of the respiration, then difference between respiration and combustion, and types of respiration, that is aerobic and anaerobic. And we also discussed the structure of ATP, structure of mitochondria, as well as pathway of anaerobic respiration, followed by glycolysis and fermentation. In the next hour, next video, we will be going to discuss about Krebs cycle, glycolysis, as well as electron transport.